Sorry. That was very, okay.
Yeah, he's the only person I'm going to know, so I'm going to stop it for a little bit to say hi to people, I guess.
We're on? Okay. Hi. So we are going to try to recompensate for the disastrous 20-minute lecture last class. So for those of you online kids, I know the VOD is being worked on to be up for last class. You probably already have realized this by the time you listen to this lecture. But just so you know, we had a power outage, which is why we only got 20 minutes of video for last class. So unfortunately, we lost a good chunk of time. So I'm working on how we can make up for that lost time, because we already have that review lecture that's scheduled for next Friday. So we may have to cancel that, but I'm working on it. So we'll just finish what we can today. I have posted your neurophysiology lectures, which would be what we should have done today and then next Wednesday. But we'll just see where we go today, and I'll make an executive decision. Okay? So I believe I ended on this slide. It was a little crazy, so I don't remember. Anybody? Okay, so we finished through the cortical lobes. I just wanted to highlight two major groups of structures that are subcortical, so beneath the cortex. And I've alluded to them already. The first group of structures that are important to highlight at this stage, and you'll return to see them over throughout the term, are the basal ganglia. And the basal ganglia are a collection of nuclei that are just below the white matter of the cortex. So you remember here, this is a coronal slice. All this gray matter is cell bodies, and the white matter is axons. So here's all that gray matter. Here's the white matter. And these collection of nuclei occur just under the white matter. They consist of three major structures: the caudate nucleus, which is here, the putamen, which is here, and the globus pallidus, which are the adjoining structures just next to the putamen. So taken together, all of these, the caudate, the putamen, and the globus pallidus, are considered the basal ganglia. Okay, so remember, yes, we have figure labeling on our exams, so anything that is in the textbook and anything that's in the lecture is fair game to be labeled for an exam. Hint, hint. So this figure is showing you this coronal section. It includes the basal ganglia, but it also includes something called the subthalamic nucleus. It's called subthalamic because it's literally beneath the thalamus here, and the substantia nigra. And these are included because these structures are also important in movement. So it makes sense that they're all located somewhat next to one another. So the basal ganglia are controlling voluntary movement. We talked about it last class, or 2 classes ago, when I talked about Parkinson's disease, which is a disease that's caused by degeneration, in particular of that substantia nigra, which makes connections to other parts of the basal ganglia. So remember, every neuron makes a connection with other neurons. These are called pathways. And so the pathways from the substantia nigra to other parts of the basal ganglia are destroyed, and this results in Parkinson's disease. Another disease, neurobehavioral disorder, that's thought to involve the basal ganglia is Tourette's syndrome. Tourette's you may have heard of. It's more, I would say, a speech or in some parts voluntary movements. These individuals have tics, and they often, you'll probably hear about Tourette's because people make fun of people with Tourette's all the time, very sadly, but they're the ones that can't stop swearing. So again, remember, frontal lobe inhibits some behaviors, and including the propensity to swear all the time, right? So that's not something we want to be yelling out swear words in the middle of church or a lecture. But people with Tourette's syndrome, due to some disconnections between these basal ganglia and the frontal cortex, are not able to control these tics. So the second important group of structures I want to talk about is the limbic system. And I've mentioned the limbic system before. It is, again, a group of structures that's located between the neocortex. Here's a sagittal slice of the brain. Remember, this is the corpus callosum. Here's the brainstem. There's the cerebellum. And it consists of a number of different structures. It always changes. What are the core structures? We always tend to include lots and lots. But the principal ones are the amygdala, which is here in purple, and it's at the end of the hippocampus here in blue. and the cingulate cortex here in green. So these principal structures are involved in the regulation of emotion and motivation, some sexual behaviors, memory, spatial navigation. I'm going to trip over this, so I will move it. Okay, so that's the limbic system. Again, we'll talk a lot more about this in the chapter on motivation and emotion. So that's it for the basic neuroanatomy. We're now going to move into more of the functionality of the nervous system. So we'll do a little bit more of the gross functionality, and then we'll move into the micro. So from macro to micro. So I talked about how the central nervous system is important in mediating behavior. We're going to talk very briefly about the somatic nervous system, which is part of the peripheral nervous system that's involved in transmitting information. So importantly, the somatic nervous system is monitored and controlled by the central nervous system. And remember, the somatic nervous system is comprised of the cranial nerves and the spinal nerves. So the cranial nerves are monitored and controlled by the brain, and the spinal nerves are monitored and controlled by the spinal cord segments. So what do I mean by the cranial nerves? Here is the dreaded diagram that every second-year neuroscience student never, ever wants to memorize, but has to. Yay! You are required to memorize the 12 cranial nerves. It's part of what everybody has to go to, an indoctrination of sorts. Essentially, we have 12 cranial nerves. They are pairs of nerves, one for each side of the body. And each of these cranial nerves exit the brain and make contacts with important other parts of the body and are responsible for a primary function. They're all given a number, a name, and a function. So for example, cranial nerve 1 here is the olfactory nerve, and it's involved in smell. Cranial nerve 2 is the optic nerve. Cranial nerve 3 is the oculomotor nerve, and it's involved in eye movements along with cranial nerve 4. Cranial nerve 5 is the trigeminal nerve. It feeds into the face, and it's important in masticatory movements, chewing, and facial sensations. Cranial nerve 6 is the abducens nerve. It's also involved in eye movements. So you can see we have 3 whole nerves involved in moving our eyes, right? Cranial nerve 7 is the facial nerve. It's involved in facial movement and sensation. Cranial nerve 8 is the auditory vestibular, hearing and balance. Cranial nerve 9, the glossopharyngeal nerve, which is involved in tongue and pharynx movements as well as sensation. So speaking, you really require a lot of your cranial nerve 9. Cranial nerve 10, it's called the vagus nerve. It comes from the word vagrant or vagal, which means traveling, because it is the one that is the most distant. So you can imagine most of these nerves hit somewhere in the face or the head. And cranial nerve 10 goes to the heart, the blood vessels, the viscera, which is essentially your guts. And it's involved in movement as well of the larynx and the pharynx. So it goes all over the place. Cranial nerve 11 is the spinal accessory nerve. It's involved in controlling your neck muscles. And cranial nerve 12 is the hypoglossal, and it's also involved in your tongue muscles. So again, speaking requires 9 and 12. So on an exam, I might ask you, what is cranial nerve 8? And what is its function? So you have to say cranial nerve 8 is the auditory vestibular. It's involved in hearing and balance. Okay? As a little competition every year, what I do is on See Learn, I will post the 12 cranial nerve competition. And your job as students is to think of some really awesome, unique mnemonics to memorize the 12 cranial nerves. So has anybody heard of any of these mnemonics? There's one in the book. It's on top of old Olympus or blah, 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 blah. So basically a mnemonic, you have to use the first letter of the name of the olfactory nerve and create a mnemonic to try to remember it, right? So there's a dirty one, to touch a female and gives, and I'm not even going to say it. Okay, so yeah, so that's a dirty one. There's some out there already that I've heard, so don't try to submit that as your own. Try and think of like one. I don't mind if it's dirty. I really don't. You can use dirty words. In fact, and I'm saying this because there's actually evidence, if anybody has read Moonwalking with Einstein. Anybody heard that book? By Jonathan Safran Foer. And there's evidence to support the fact that we remember things that are shocking, right? And so people who enter into these world memory competitions where they have to memorize a stack of cards, for example, in the order, like a jack of hearts, queen of spades, etc., they use dirty imagery that they pair with the specific card to remember the order of the cards. So I don't care if it's dirty. I'm not going to be like, you've heard me drop the F-bombs. I'm not going to be insulted. One that won last year actually had a bestiality theme. So, again, I will, and what I give you guys is a $5 gift card to either Starbucks or Tim Hortons as your prize. Okay, so Ren.
I'm just wondering, for the exam, will we have to, like, memorize the locations of them, like, on the picture?
Quite possibly, well,
like,
you mean
fill
like...
in the blanks or whatever?
I mean, that's a really arbitrary diagram, so I don't think I would do that to you. I don't know. Like, blank out 8 and say, which one is this? Yeah, no, I wouldn't do that. No. Okay? So you have to memorize your 12 cranial nerves, but fun competitions will ensue. And you guys online, absolutely, please participate, because I've noticed a lot of you aren't participating. What's going on? Okay, moving right along to the spinal nerves. So we talked about the cranial nerves, which is controlled by the brain. The spinal nerves are controlled by the spinal cord. These are both part of the somatic nervous system. And the spinal cord is essentially, it's a string of 31 pairs of nerves that go all along your back. And I've talked about it previously, I believe. And it makes connections with the somatic nervous system. You recall, I can't remember, did I teach this already? I feel like I did. Yeah. The dorsal fibers are the ones that are afferent. So they carry information from the body's sensory receptors into the brain. So every spinal cord segment has a dorsal. So remember, dorsal means towards the spine, and a ventral towards the stomach, or top and bottom. It's up to you how you can think about it. And the dorsal fibers are the ones that carry information from your sensory systems. And the ventral fibers are the ones that are efferent, or they exit. And they come from the brain, they go down the spinal cord, and they exit that part of the spinal column to make contact with that part of the body to move it, right? So we talked about this earlier. If you touch your finger to something hot, those sensory those neurons that have their fibers on the tip of your finger are going to travel up your arm into your spinal cord, into that specific level of the spinal cord that controls your hand. So you can see here it would be part of your cervical spinal cord right here, because that's what controls your finger. And then it would go up into your brain. Your motor cortex would send a signal back down, so the exiting fiber to make connections with your muscles to move away from that hot plate, okay? So yes, this is you can have a sensory reflex where you don't involve the brain, because in fact, if it's something hot, it's literally just one interneuron. That means that you move it away, but it does eventually get to your brain. What's fascinating is that back in like the 20s or 30s, these two scientists that actually they hated each other. They were working down the hall from one another at a university, and they both came to the same conclusion around the same time. And they both came to the conclusion that the general principle that the sensory fibers are located more dorsal and the motor fibers are located ventral. And this continues all the way up into the brain. That we have this segmentation of sensory and motor information, where sensory information is more dorsal and motor information is more ventral. And this is now called the law of Bell and Magendie. So that is that law that, generally speaking, in the nervous system, dorsal is sensory, ventral is motor. Each spinal nerve makes contact with a very specific part of your skin. And this is called the dermatome. All right, so all over like the surface of our body, you can imagine is a map of individual dermatomes. And so that's the area of the skin that is supplied with those afferent nerve fibers by a single spinal cord dorsal root. And it makes sense that the adjacent or neighboring regions of the spinal cord are working together, right? It doesn't make sense that one part of your spinal cord controls your foot and the neighboring region controls your finger, right? You'd want your fingers, each of your fingers, to be represented close to one another in the spinal cord, and then your arm, your forearm, your upper arm, and your neck, for example. So that's essentially what this diagram is showing you, is that all along the cervical column here, you have the different nerves, so the cervical nerves, thoracic, lumbar, and sacral, that control neighboring dermatomes, right? So these cervical nerves control your head and your arms and your shoulders, thoracic nerves, your thorax or your trunk, the lumbar nerves, your waist region, and then the front part of your legs, and the sacral nerves, as well as the coccygeal segment, which controls your back part of your body. Is that clear to everybody? Rachel? Rebecca? Rachel.
You were saying before how there's like the map of the nerves.
So I was saying before, on the somatosensory cortex, we have a map of the human body. Yes. Yes, exactly. So we'll learn about, well, we may not actually learn too much about this, but Wilder Penfield was the one that found that, yes, every single point on your body is represented, there's a specific cell that fires in your motor cortex and in your somatosensory cortex in response to that being touched. And what's happening is that signal is being sent from the dermatome up into your arm, up into your spinal cord, into that appropriate region of the spinal cord, and then up into your brain. And that is, the map is conserved. Right. More or less, except for, remember, the genitals are next to the feet for some reason. So a little bit more about the autonomic nervous system, because the autonomic nervous system is responsible more for balancing of the internal functions. So somatic nervous system, movement and sensation. Autonomic nervous system, digestion, heart rate, respiration, etc. So remember, there are two divisions working in opposition. The sympathetic division of the nervous system is the one that's involved in fight or flight. So in the face of a real or perceived stressor, this is a system that activates the organism to deal with the stressor. The parasympathetic nervous system, which is the opposite side of the seesaw, is the one that's the rest and digest. And it's opposite. Remember, it reverses the fight or flight response. This is also known as the 4 Fs: fight, flight, feeding, and sexual reproduction. So it's basically, again, activating the organism. And the parasympathetic is the one that traps energy. It's resting and digesting. And it also is responsible for shutting off the fight or flight response. So different group of nerves that are attached to the same regions of the body. And you'll see this in this other image here, where we have the parasympathetic and sympathetic divisions. So here, this is the stimulatory fight or flight. And again, they come out of the spinal cord and the different regions of the spinal cord here. These ones, the parasympathetic, they're coming from the vagal nerve, the facial nerve, and then other parts of the spinal cord. But importantly, remember what I was saying is that they will all make contact with the same organs, but it's different groups of nerves, because those different groups of nerves are having the opposite function. So whereas here in these nerves are dilating your pupils during the fight or flight response, the oculomotor nerve will contract your pupil. So it's the opposite response. Here, this nerve will inhibit salivation. This one will stimulate salivation. This nerve will relax the airways. This one constricts, inhibits digestion, stimulates, et cetera, right? So you've got these opposite functions different groups of nerves that are controlling that. So there's different, you can imagine, different signals that are arriving at this organ to initiate that function or response. Okay? It's a pretty busy diagram, but as long as you're able just to say each of these is controlling the same body system but having an opposite effect, you should be okay. Don't worry too much about memorizing what is the sympathetic chain and this stuff, like where it's emerging from. I don't expect you to know that. I just want you to know the basic principle that they work in opposition for one another, and one's fight or flight, one's rest and digest. Okay? Yeah?
Do we need to know, like, to any extent, like, the actual changes that happen in the different body parts, or do we just have to know that they're different? Like,
No, I would expect you to say, okay, when we're talking about digestion, what would happen in a fight or flight response? Yep. Ren?
Just out of curiosity, the very last one. So for the fight or flight response. Okay, just answer it then.
So why are we getting ejaculation in a fight or flight response and an erection in a rest or digest? I know there's a reason. I just can't think of it right now. It's funny, sometimes, like in the lab, you'll see this, that in rats that we, for one reason or another, end up in that fight or flight response, and they have ejaculation responses, which we don't tend to see in higher organisms. So I think this is inhibited in humans, right? Because I don't think anybody's experienced this. But I'll look into that. It is curious. I was looking at that and I was thinking in my head, someone's going to ask me and I don't know the answer. So I'll find that out. All in the name of science. As the camera zooms in on my face. All right, so moving away from, again, gross neuroanatomy and then the very simple functions of the somatic and autonomic nervous systems. And now we're going to go really much more micro, and we're going to look at the units of the nervous system, which are the cells. We're going to look a little bit about the internal structure of a cell, and then we're going to talk a little bit about genes, cells, and behavior. Just a FYI, I removed a lot of the stuff on genetics from my lecture. It is in your textbook, and you are required to know it, but I didn't want to go through it yet one more time because I feel like it's so review. So what I did is I gave all my lecture slides to the TAs for that review lecture that is being planned for next Friday. I'll keep you posted as to whether that's still going to happen. So for those of you that don't have a strong background in science and you're like, genetics, huh? I would strongly recommend that you come to that lecture if you need a bit of a primer on how we make genes, okay? So I removed that from my lecture today, but it will occur either in a review lecture that'll happen on Friday or at another time. All right, so we all remember from Psych 100, hopefully, that the basic unit of cell function in the brain is the neuron. We also have glia, and we'll talk about those shortly, but we will focus on the neurons because these are the ones that do the main function, which is to communicate with other neurons. So what you should always be able to do in an exam and otherwise is identify the main parts of a neuron. So the dendrites here, which are the part of the neuron that receives information, are connected to the cell body, or the soma, which integrates information, and the soma is connected to the axon, which sends out information. That's the basic parts of a neuron. You should always be able to identify them. Dendrites gather, cell body integrates, and the axon carries the information to be passed on to the other cells. Are we all good? Is this like snooze? Yes, awesome, good. If not, I have my handy dandy, somebody knit this on Etsy, wicked neuron. It's a very specific kind of neuron. Does anybody know what kind of neuron it is? I'll give you a clue. 3 basic neurons: unipolar, bipolar, and multipolar. It's a bipolar neuron, yes, right? Because it's got 2 main poles here. So yes, very fun. I will use this throughout the day. It can also be used as a choker, should you want to have some fun. All right, so again, now that we have the basic understanding of the parts of the neuron, we need to delve even deeper because we're going to be talking later on about how the cells communicate. So one thing that you should know is there are specialized structures all along the dendritic tree. So each of those dendrites, it's like a process that extends from the soma. And there's little specialized structures that are called dendritic spines that protrude from the dendrite. And the whole purpose of having these spines is they act to greatly increase the surface area of the dendrite in order so that it can receive a lot more information, right? So you can see here, this is a cell body and the dendrites. And you can see these little protrusions here are the dendritic spines. And you can see these are the axon terminals of an incoming neuron that's sending its signal. So it's making connections specifically, not always, but sometimes, mostly, with these dendritic spines. Okay? Another important part of the neuron is the axon hillock, which is the point at which the soma makes contact with the dendrite or axon, in this case of the bipolar neuron. So it's usually like a triangular portion that emerges out of the cell body. And we'll learn a lot more about the axon hillock because it is a very important function in the transmitting of information when we talk about neurophysiology. So as it says in the slide here, it's where the action potential starts. So the other parts that you should be aware of are axon collaterals, which are not shown in this image. But essentially, even though, like in this, well, I guess, yes, these are axon collaterals. So you can see there's one axon that emerges out of the cell body here. But down here, you can see this branching. That's the axon collateral, right? So lots of different processes that spin out of the one axon. Where did my... All right, in addition, as the axon gets towards the base, so the bottom, you have what are called teleodendria. And these are the very end branches of the axon. And at the very bottom, which you can see in this picture, you have what's called the terminal button. So the axon essentially branches out to form the teleodendria, and at the very tip of each of these teleodendria is the terminal button. And this is the one that, this is the part of the neuron that is involved in the synapse. Right? So we've heard about the synapse before. Here in this image is a blow-up of that synapse, which includes the end button, sometimes called the end button, terminal button of the sending neuron, and the membrane of the receiving neuron. It's also called the end foot. Questions? Not too many things that I'm playing with. So the synapse, you've heard it before, is that gap between one neuron and another neuron. What you'll learn next class is it's not just, you've learned before, I'm sure, the synapse is that little space, right? Everybody's heard that? It's a space between the neuron that sends and the neuron that receives. In fact, that space is called the synaptic cleft. The synapse includes the pre- and the postsynaptic bits, right? So we'll learn a little bit more about that next class. So it's usually between the end foot or the terminal button of the axon of one neuron and the dendritic spine of another neuron, as you can see in this image here, the end foot and the dendritic spine here. I feel like I'm going fast. Am I going too fast? Yes? I will slow down. It's like in my head, we lost so much time. Take a moment to pause. Any questions? Okay. So as I mentioned, the main function of the neuron is to receive information and to send information out. What exactly that information is and how it does it, we do it next class, or potentially this class if we have time.
Yeah,
Yes.
I had a question back to the axons. One of them is the end
branches of the axon. The other one is the branches of the axon. Like, what's the difference between
so,
the axon
like, this is not the
collateral?
greatest. I shouldn't have used my crazy knitted thing, but let's go back to the board. Here's our cell body. Lots of branching. So, in fact, you have, these are the axon collaterals. This is the teleodendria. And at the end of every teleodendria is that terminal button. Okay? So sorry, I did not make that very painfully clear. I think there might be a diagram in your textbook that might explain this better. But art. Yes?
So then the next neuron is going to be like the dendrite at the bottom where the teleodendria is.
Yes, so this, let me just add another one so I can make it even more clear, is going to make connections with the dendritic spine here. So that would be the synapse. This is yet another neuron. And it keeps going and going and going. Yeah. Okay? So, this is again from your textbook. And what you can see is that, again, the information flow of the neuron is such that the dendrites collect the information. So each neuron gets inputs from thousands, potentially thousands, most likely hundreds of other neurons. So they come in via that, those messages are received via the axons of all those other neurons, which converge onto that one neuron. It's collected at the dendrites. The cell body integrates all that information. So again, what we're going to learn in next class is what do I mean by integrate? Well, what does that exactly mean? At this point, I can't go into that much detail because I'll confuse you all. But just know that the information is integrated because every neuron, it's getting some information that's saying do this, other information saying don't do that, some information that's saying no, don't do the first thing, do this other thing. Right? And what the cell body has to do, for lack of anthropomorphizing, is it has to make a decision. Which information is the strongest? So what is the signal I should actually send? So the information is sent out via the axon, and then it gets to the terminal buttons, and then it's passed on to yet another target. So it's really hard to explain these things in isolation because our brain doesn't work on a one neuron level. Our entire brain is humming away, receiving thousands of inputs and sending out thousands of inputs. So it's really hard to extract it down to one neuron sending out one piece of information because that's a very artificial situation. Okay? So before we get into more functionality, you should know that there is different types of neurons in the nervous system, and we can classify them according to their structure or their function. Here, I've classified them according to their function, and you should kind of be cued to this already, that there are specific neuronal populations that we consider to be sensory neurons, which means they bring information into the central nervous system. Okay, so this, the example this is from your textbook, this is a somatosensory neuron. So it receives information from your skin or your muscles, and it sends that up into the brain. In addition, most of the neurons in our brain are what are called interneurons. So they are the gaps between sensory and motor, because you'll see that the third kind are the motor neurons, because these are the neurons that send information out and actually enact movement. So the interneurons associate sensory motor activity within the central nervous system. Here are some examples of different types of interneurons. A stellate cell, which is found in the thalamus. A pyramidal cell. It's called pyramidal because it's triangular shaped like a pyramid. So it's based on its actual, what it looks like. And these are found in the cortex. And then this is a Purkinje cell, and it's found in the cerebellum. So different kinds of neurons that do different things. Here is that motor neuron, and you can see that based on what it looks like, it is quite different and unique compared to the sensory and interneurons. Basically, its dendritic tree is what is different amongst all of these. Ren?
So just to confirm that would make sensory neurons afferent and motor neurons efferent?
Yes, okay, that's right. Okay, so we're going to move along to looking at glial cells, which I introduced a couple classes ago. And these are the kinds of cells that they're more numerous. There are more of them in the brain compared to neurons, but they do a very different role. They primarily support the nervous system. They have a supporting role. And so I'm going to go through 5 different kinds of glial cells that we have in the brain. The first are the ependymal cells. And this is a picture that I found on Google image, just showing you that the shape of these cells is small. They're ovoid, meaning kind of like egg-shaped, and they're found in the walls of the ventricles. So remember, you have 4 ventricles in the brain, the lateral, 2 lateral ventricles, third ventricle and the fourth ventricle. They circulate cerebral spinal fluid. So in fact, what the ependymal cells do is they make and manufacture cerebral spinal fluid. So that's their main role. Anybody heard of hydroencephalus? Or hydroencephalus. So in some cases, what happens is that if a baby is developing in utero, sometimes you can get a block somewhere in the ventricles. And what that does is it prevents the cerebral spinal fluid from circulating normally, and you end up with a buildup of cerebral spinal fluid in some part of the brain. This is very, very bad. If it's left untreated, you can develop mental retardation because what happens is it builds up the pressure in the brain, and all the cells that are overlying that don't get normal functionality. So here's a very creepy picture. If you're kind of scared of aliens, which don't exist, but whatever, you may not want to look at this. But this is an example of an infant who had hydroencephalus and did not survive much past birth. But you can see how large the head is due to all this increased cerebral spinal fluid. Here are photographs that show the brain of a 10-year-old child who had developed hydroencephalus in utero as a result of a block in the cerebral aqueduct, which connects the third and the fourth ventricle. And you can see how large the ventricles are in relation to the rest of the brain. Nowadays, we can detect hydroencephalus, and it is easily treated with a shunt. And what you do is you surgically implant this shunt into part of the ventricles, and it diverts the fluid away from the block and remaps it, I guess, for lack of a better expression. So it is treated. Were there 2 questions? That was just you were going to ask. Yes, there you go. I knew it. All right, so the second kind of glial cell is something known as an astrocyte, and I believe I talked about them a couple classes ago. This is a figure that's reproduced from your textbook. They're called astrocytes because they are star-shaped. That's the word astro. Cyte means cell. Anytime you see C-Y-T-E, that just is Latin for cell. So star cell. They're also symmetrical, so they have equal number of processes on each side. And they provide a lot of structural support for the neuron. You can see here in this image the neuron that's shown, and the astrocyte nearby that makes contact with it. Importantly, what it does is the astrocyte also has end feet that make contact with the capillary walls of blood vessels. So what they're doing is, remember, everything in your body is transported via the bloodstream. So important nutrients, oxygen, glucose is in the bloodstream. And what the astrocyte does is it carries these important nutrients and brings them to the neuron. Okay, so it transports these substances between the neurons and the capillaries, and also is important in protecting the brain, which it forms what's called the blood-brain barrier. We talked about the blood-brain barrier. I feel like I should have. So the blood-brain barrier, all throughout your, all the blood vessels of the rest of your body, the walls of the capillaries, which are the the capillaries, which are the cells that line the walls of your blood vessels, are very tight together. So here is an example of a blood vessel. These are the capillaries, so the cells that line the wall of your blood vessel. And they're tight junctions, which means nothing can go into, they're all throughout the rest of your body, there actually is more holes, so not as tight, right? So stuff can flow into and out of the bloodstream. The cells, the blood vessels that line your brain, remember we talked about blood vessels a couple lectures ago, the major cerebral artery, etc. The capillaries are tightly packed, which means the spaces in between them are really, really small, so very little can flow into and out of the brain. And part of what the astrocytes do is they control for that as well. They are also involved in scar tissue formation, so they have a role with the immune system, because if you have damage to the brain, the astrocytes will come in and help repair the brain by forming scar tissue, and they provide the fuel. So major supporting role. Another thing that they do, as you can see from this diagram here, is they provide myelination. So their end feet will wrap around the axon of the neuron to which it is connected and provide that sheath that covers the axon. And you'll learn next class the important role for myelin. So another kind of glial cell is something known as microglia, small glia. They originate in the blood because they are an offshoot of the immune system. So microglia's main role is something they do is called phagocytosis. They literally will eat off dead cells. So once a cell dies in the brain, yum, 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 the microglia will go down and eat it. So you can see the brain is very environmentally friendly. It picks up its own litter. Fourth kind of cell is something known as the oligodendroglia. And this is a kind of glial cell that's found only in the central nervous system. And it is also involved in myelination. So it's oligodendroglia, only in the central nervous system, and its primary role is to provide myelin. So remember, it coats the axons. By contrast, the final type of glial cell is something known as a Schwann cell. It is like the cousin of the oligodendroglia. So whereas the oligodendroglia myelinates cells in the nervous system, the Schwann cell myelinates cells only in the peripheral nervous system. So oligodendroglia, central nervous system, Schwann cell, peripheral nervous system, myelin. That's what they do. So the one difference between Schwann cells and oligodendroglia in terms of myelination is that with Schwann cells, there's a one-to-one relationship between the glial cell and the neuron, meaning only one Schwann cell will myelinate only one neuron. And with oligodendroglia, one oligodendroglia might myelinate multiple neurons. If anybody's heard of multiple sclerosis, or MS, this is a neurodegenerative disorder. It's considered an autoimmune disorder, so women are more susceptible to it than men. And it's considered autoimmune because the body attacks and considers its myelin sheath as being a foreign substance. So autoimmune, self-immune. And so with people who have MS, they have a loss of the myelin sheath of their peripheral nerves. And what that results in is, because myelin is important in sending that signal from the cell body down to the end foot, if you have lost that myelin, the signal is sent much slower. And so people who have MS, they have pain, they have a hard time with movement, because remember, it is that peripheral area. Yes?
Going back to the oligodendroglia, does it have the same nodes of Ranvier like the Schwann cells do, like a myelin sheath?
Okay, you're getting ahead of yourself with the nodes of Rognier. That's fine. I appreciate that. But we'll talk about that next class. Yeah, no problem. But essentially, all of myelin can produce that these little nodes. Okay? Yep.
For MS, does that mean that there's absolutely no myelin, or is it like there's some, but
It's because it depends on the kind of MS. So whether it's relaxing remitting or I forget the other kind, not my area. Yeah, and it can because it's progressive. It can eventually attack all neurons. It can be quite bad. There was a student I taught a couple years ago who her MS had degraded her optic nerve, so she lost her vision. So it can affect people differently. So some myelin can be conserved in your arm, for example, but you've lost the myelin in your legs. Yeah. Okie dokie. Perfect. Yes.
Back to the astrocyte.
Yes.
Does it provide myelination on top of an already myelinated?
Yes. Yeah. So the astrocytes are also capable of myelination. Yeah. Anybody else? Yes. Rachel.
Does the myelination of the astrocytes the same for all as an effect as
Yeah, so anytime you myelinate, the whole point of myelination is to increase the speed of the signal. Yeah, so we'll get to that a lot next class. Yeah.
So the glial cells aren't involved in information transmission at all?
So are the glial cells involved in information transformation? Up until the last 10, 15 years, it was thought that no, but now there is increasing evidence to support the fact that the glial cells do have communication properties. As yet, it's not really well understood, but there is some suggestion that they can communicate with one another. So what happens when we have damage to our cells? Well, we've touched on the idea of paralysis, and essentially what happens is that if we have damage to cells in the central nervous system, we cannot regenerate those cells. Okay, so cells that die off in the central nervous system, we cannot regenerate them. Remember, the spinal cord is part of your central nervous system. So cells that die in your spinal cord, or if your spinal cord is severed, we cannot, at present, regenerate or make connections in the spinal cord with these axons. So what happens is that if you have cells that die in the spinal cord, you have what's called paralysis. And it's loss of sensation and movement that's due to injury in your spinal cord. You can either be quadriplegic or paraplegic. Remember, quadriplegic is the higher up that the spinal cord is severed, the more likely you are to lose capabilities of your arms and your legs. If it's lower down, it's just your legs. So repair does not take place in the central nervous system, and regrowth may even be inhibited. That said, there's lots of money being poured into labs that look at spinal cord regeneration. Christopher Reeve set up a whole foundation before he died, and he was very generous with his money in putting that towards research that looks into spinal cord regeneration. If any of you are interested in this topic for a future honors thesis, for example, Patrice Smith, who is one of our faculty members here in the Department of Neuroscience, this is what her lab studies, is spinal cord regeneration. In the peripheral nervous system, however, what you may have noticed if you've ever had, if you've cut yourself somewhere in your arms or your legs, severely enough to actually sever your nerve, what you may have noticed is, oh, wonder of wonders, the nerve can actually regrow. So when I was in my 4th year of my undergrad, I was doing surgery on rats, and I was changing a scalpel blade. And I'm left-handed, so I'm quite clumsy. And I accidentally, the scalpel blade, as I was changing it, it snapped and embedded into my 2 forefingers on my left hand. So in fact, I'm usually fine. Because I'm so clumsy, I'm all good. I'm fine. Blood, it's all fine. So I took it to, there's a master's student in our lab who had an MD from Syria, and I was like, Maria, I think I might need stitches. Can you stitch me up? Because we had all suture kits in the lab. It was all good. And she looked at my fingers, and she was like, Kim, no, you've severed potentially your tendons. You've got some nerves that have been severed. We need to take you to the ER. And then I was like, oh, dude. And then I got all faint, and I was not in a good place. So we took me to the ER, and sure enough, I hadn't severed the tendon, but I had severed the nerve. And so they stitched me up, and we had a plastic surgeon come in and reattach the nerves. And so what happens when you do this is that initially, like, I couldn't bend my fingers. But over time, what happens is these nerves regrow, right? So you have the microglia and the Schwann cells that help repair the neurons. And here in this handy-dandy slide, you can sort of see what happens is that when this peripheral axon is cut, the axon will die. What happens is that the Schwann cells will first shrink and then divide, and it will form glial cells along the axon's former path. So the neuron will generate these sprouts, and then the Schwann cells will line up along where the axon used to be, and then they'll reform. So they envelop the axon, and they will form new myelin. And when the nerve is regrowing, what you may have had experienced, if any of you have had this happen, is the area feels really itchy. That's one of the signs, it's actually a good sign, showing that you're getting regrowth. And they don't know why this itchiness is happening, so don't ask me. But essentially, this is a sign that you're getting regrowth. And eventually, I had to do all this like physio with one of these balls, and it really sucked because I'm left-handed, so I couldn't use my left hand for a while. But there you go. Crazy story about rats and scalpels. Rachel.
Are there any theories about why
the peripheral nervous system regrow and the central nervous system does not? Not that I know. I can look into it. Yeah, so why do we have this system in place where our periphery can be repaired, but our central nervous system cannot? Don't know. Anyone? PhD thesis? Sold. All right, so moving along to the last portion of this lecture, remember that I took out all the slides that I had in advance of this, which was here's how genes are manufactured and all that messenger RNA and transcription and translation. So if any of that just coming out of my mouth made you feel like, what is she talking about? It's German Shepherd to you. Again, you will definitely need to read the chapter in your textbook and attend the review lecture. Because I'm going to move away from how genes are made to more of their functionality, right? So we know that genes are formed of make proteins, which are the building blocks of the body. If you don't know what a protein is, yowza. So proteins are also important in making cells, and our cells are important in mediating behavior. So it's important in a class where we're learning about biological foundations of behavior to understand that genes are really one of the most foundational pieces of our behavior, right? So a lot of what we do is based in our genome. Not necessarily in learning, but in our genome. So a little bit about genomics, which is the field of study that looks at how genes produce proteins. And if any of you are tuned into this, you know that the human genome was finally identified after a long period of time. So we have an idea of all the genes that the humans manufacture, but we don't really know what they do. And proteomics is the field of study that is directed at understanding what all of these things do. So we know all the genes and the proteins, the products that they make, but we don't know all of what they do. So proteomics is that field of study. Interestingly, genome size and chromosome numbers seem to be completely unrelated to organismal complexity. And this is a table in your textbook, table 3.2, which really shows like basically worms compared to humans and how there's really no relationship between chromosomes and genome size, which is kind of curious. So a little bit more about chromosomes. This should be a review for anybody who has had biology as a background, and I apologize. But we do need to make sure that everybody else is on the same page. So humans, our somatic or bodily cells, have 23 pairs of chromosomes. When we are formed, at the moment of conception, when mom's egg meets dad's sperm, what happens is we get some of dad and we get some of mom, right? And what of mom and dad we get is kind of a crapshoot. But essentially, every chromosome has one copy from their mother, the X, and one copy from the father, right? And so these are chromosomes 1 through 22. The sex chromosomes, which are pair number 23, this is what determines whether you're a male or female, right? So what is this person on this figure? Male. Okay, is anybody like, I don't know what you're talking about? Okay, so XX, female, XY, male. Okay? So this person is clearly male. So chromosomes are where the genes can be found, right? So here you can see that alleles are alternate forms of that gene, right? So here we have a pair of chromosomes where the gene pair contains 2 alleles, right? So for example, in this red one, we have this allele here, BB, this allele here, AA, and this allele pair here, PP. When we have the same allele, or 2 identical alleles, this is known as something called homozygous. So if you're homozygous for something, in this case, P and A are homozygous. They have both the capital P and both the small a. The allele B is heterozygous. Homo means same, hetero means different. So big B, little b. Okay? So if your mother has brown hair and your father has brown hair, and both of them have dominant, 2 dominant copies of that gene, you are going to have brown hair, and you're going to have a homozygous allele, right? If you have, in this case, big B, little b, heterozygous, you can say that's the gene for brown hair, but brown hair is a dominant gene compared to blonde hair. So you would have brown hair, but you'd have a heterozygous allele. Okay? So you can have brown hair as a homozygous or heterozygous situation. So again, what I mean by dominant and recessive, so first of all, before I get to that, when we study in genetics, often we use like mouse models to look at genetic mutations. We often refer to the wild type or the mutated copy of the gene. So wild type, if I'm referring to wild type, it refers to the normal phenotype or genotype. So wild type is the normal, the baseline. By contrast, the mutation is the gene that we've screwed around with, right? So this is the alteration of the allele that yields a different version of that allele. Okay, so we have the wild type or mutation. The genotype is what your genes are. The phenotype is the expression of those genes. Okay, so genotype, big B, little b, phenotype, brown hair. Okay, genotype, little b, phenotype, blonde hair. Genotype, whatever the hell gene it is that makes you express green eyes. Phenotype, green eyes. Okay? So the phenotype is the appearance that results from the interaction with the genes and sometimes the environment. Okay? So most of what we are as humans today is in our genome, right? So what color our skin is, our hair, the size of our lips, the size of our nose. A lot of our behaviors can be determined by our genotype, and this is why we're talking about it in this context, right? Because even in biology classes, we talk about bodily genes, but in this class, we're thinking about behavior. So we have to understand that a lot of our behavior is controlled by our genes. So again, what do I mean by dominant and recessive? So if you have alternate forms of a gene, so different alleles, you can have the allele that's either dominant or recessive. So the dominant is the member of the gene pair that is routinely expressed. So what that means is that if you have a heterozygous inheritance of these alleles, the dominant gene is the one that's going to be expressed, right? So little b, big B, if the big B codes for brown hair and the little b codes for blue hair, blonde hair, If you end up with brown hair, then we can assume that the brown is the dominant gene. Okay, again, this is all like routine information from biology grade 11. The recessive allele is the member of the gene pair that is routinely unexpressed. So, for example, blonde hair in a population is a recessive gene, which is why it is not as common, and some would argue more highly desired. A lot of women tend to dye their hair blonde because it makes them unique or different. So we can think about dominant and recessive genes in the context of neurobehavioral disorders, right? Because there are some neurobehavioral disorders that can be inherited. Things like Huntington's disease, for example. So an important thing to realize is that with dominance and recessive, you don't always show the full dominant gene. That would be what's called complete dominance. So brown hair, for example, is a form of complete dominance, where only the dominant alleles trait is expressed in the phenotype. Incomplete dominance is when the phenotypic expression of the dominant alleles trait is only partial. So, for example, if you have wavy hair like I do, that's considered incomplete dominance because you only have partially of the gene expressed for curly hair. Okay? Everybody understand that? So wavy hair is incomplete dominance. Dominant hair, I can't remember. I think it's straight hair is dominant, curly hair is recessive, but you're getting partial. Actually, no, it would be the opposite. Curly hair is dominant, straight hair is recessive, so you're getting partial dominance of that curly gene being expressed. Yes?
So would like a speckled hen be incomplete, or would it be codominance? There you go, okay.
Another thing you can have is codominance, and this is when you have both of the gene pairs expressed, right? So, for example, this is a roan cow, and you're getting both the orange hair, it's not fur, it's hair, and the white hair that's being expressed at the same time. Anybody think of any other expression that, any other examples of codominance? So yes, the speckled hen, yep.
I think I'm just guessing here. I have someone in my family, she has one eye green and the other brown.
Yep, that's codominance. Yep, so when you have different colored eyes, and huskies are quite, have this propensity to have this. Ren?
I was just going to say heterochromia, and then she said.
There you go, heterochromia. It has a name.
Is it possible to have a codominance even if one strain is dominant over the other? Like, isn't brown eyes more like dominant over blue eyes? Like, how would you have one blue eye and one brown eye?
But that's because it's codominant, so you're only getting the expression of brown eye in one eye at the same time as the other eye is green or blue. Yep.
Is dirty blonde hair codominant or like
Dirty blonde?
I will have like a mix of brown and blonde hair?
Potentially. I'm not a geneticist, so je ne sais pas. Yeah.
Just about the eyes being different colors, what happens when someone has, for example, I know someone who has one blue eye and the other eye is half blue, half green, right down the middle? And then what happens in that case, or is that just it?
Freak of nature?
Yeah.
Just kidding.
But it's like
Haha.
straight down the middle, like very obvious.
Ren,
region.
seeing as you have a knowledge of heterochromia, is that the case of heterochromia, or when it's the one eye?
I think it can be in both or just one. I know somebody that has one eye that's half green, half blue, and the other one's like blue. So
Potentially.
that's the same thing.
Very curious.
I have to know.
Invariably, people ask me questions in this lecture related to these kinds of things, and I never know the answers, so I'm sorry. People always want to know, what about red hair and freckles, green eyes? What if my earlobe is attached on one side and not on the other? Sorry. But I will investigate. So as you know, genes can mutate, right? This is how we have change in the populations, okay? So sometimes these can be good things, right? If we mutate to have a gene that makes us really, really strong as a male, this is pretty good. But sometimes it can have negative effects, right? The effects can be specific or they can be widespread. So they can be a gene that's mutated in one part of your eye, or it can be a gene that is mutated that controls for the expression of your skin, for example. And something like alopecia might arise, where alopecia is you have no body hair, right? So no eyebrows, no eyelashes, no hair anywhere. So it can be specific or widespread. I'm going to give you an example of two kinds of diseases that we see as a result of a genetic mutation. One of them is something known as Tay-Sachs disease. And it's an inherited disease. It's caused by the loss of genes that encode an enzyme that's normally responsible for breaking down fatty substances in the body. So you can see here, it's a recessive gene. Here's the normal allele. Here's the Tay-Sachs allele. We have this is a person who's called a carrier. They don't express the disease because it's recessive, but they are carrying the gene copy. And then they mate with a normal person. And what happens is then you have a 50-50 chance of having just a normal child or an offspring that is a normal carrier. However, what happens is, because it's a recessive gene, if two parents mate, each of them is a carrier, then you have a 1 in 4 chance of producing a child or an offspring that has Tay-Sachs, okay? Because you have a 1 in 4 chance of it having both of the recessive genes. You have a 50% chance of it being a normal carrier, and again, a 25% chance of it being normal. So it's a very devastating disease. It appears around 4 to 6 months after birth. Because you're not breaking down these fatty substances in the body, they are building up, and this can have drastic effects. It results in mental retardation, a lot of physical changes, because you can imagine you don't have good motor control. And usually the child does not survive past the age of 5. Very, very sad. Anybody know about Tay-Sachs? Yeah, okay. Any, people who have it? Yeah? Yeah.
I learned in my developmental class, my professor said that it's almost exclusive to the Jewish population.
Right.
To do with their heritage or genetic makeup.
It's probably because, within Jewish populations, they tend to marry and have children together. And so as a result, if you have one copy of a gene that's being carried, and you're more likely to have that occur and proliferate if only that population is mating with each other, right? So it probably wouldn't happen as much if they interbred. And I hate to use that very crude word, but that's essentially what happens. Right, so there's a lot of diseases that tend to be found in very specific ethnicities. So type 2 diabetes, for example, sickle cell anemia. Greek people are very susceptible to this blood disease as well. So it does happen because you're staying within the same breeding pool, again, for lack of a cruder expression. So Tay-Sachs, very sad. So that's a recessive allele. Here's what happens if you have a dominant gene, right? So an example of that is, as I mentioned, Huntington's chorea, which is not altogether dissimilar from Parkinson's disease in that it is a neurodegenerative disorder that results in problems with motor function. It's an autosomal disorder, meaning it's not carried, it's not sex-linked, it's not carried on the X or the Y chromosome. It results in these motor and cognitive disturbances. So let's say, for example, we have, because it's a dominant gene, it's going to have a different pattern of inheritance. So if a carrier mates with a normal individual, here you see the increase. We have a 50% chance of that person having Huntington's disease, right? If two carriers mate, it's increased to 75%. So very devastating disease, very devastating. And it's, yeah. So it's caused by, we know, this is one of the neurodegenerative disorders. We know what the etiology or the cause is, which is an increase in the number of CAG, which is cytosine, adenine, and guanine, which are the different amino acids that repeat on chromosome 4. Okay? Oh, sorry, amino acid bases. Rachel?
How is it that diseases such as this get passed on? Because, like, Tay-Sachs died from their blood, is it going to be able to pass those genes on?
Because you can survive as a carrier.
And it just lasted this long.
Right. Yeah. Yeah.
Doesn't like Huntington not start to
Show up until your late, mid to late adulthood. which is also why it's devastating, because until we had gene therapy, people didn't realize that they carried the gene. Right now, you can send away for a kit and do your saliva and send it away, and you can find out if you have Huntington's. But what would typically happen is, somebody would have it, they'd marry, have children. The age of 35 or 40, they'd start to show their symptoms, and then they'd be told devastatingly that their children had a 1 in 2 or 2 in 4 chance of 50% chance of having it.
And then another question with Huntington's, is that because like the age expectancy, as soon as they increase a lot, so the last.
Yeah, so some theories of why we have more of these genetic mutations is because we're increasing our lifespan. So we're seeing some of these things that are popping up later in life than we would have normally seen if we had died at the age of 35. Yep. All right, so the last thing I want to talk about, one of the other chromosome abnormalities that I'm sure you're already aware of, things like genetic disorders that involve aberrations of part of the chromosome. So in this case, you're not getting a mutated copy. You're getting an actual problem with the chromosome rather than a single defected allele. Anybody know what's up here on the screen? Yeah. Trisomy 21, also known as Down syndrome. And this is essentially due to the fact that that person is born with an extra copy of chromosome 21. And what this results in is it can be on a spectrum of mental retardation, so you can be high functioning, low functioning, and a number of other abnormalities. You can see people who have Down syndrome have a very specific facial disformities. They're also shorter in stature. They tend to have problems with heart disease early on, and their life expectancy is a lot shorter. A lot of them can live very amazing, fruitful lives. It is more populous in older women. So usually if you're having a child over the age of 35, you get tested for Down syndrome because as you age, you're more likely to produce an offspring that has Down syndrome. So a little bit, the last thing we'll talk about is genetic engineering. As if you guys are neuroscience students, perhaps if you're BSc Psych students, you'll learn a lot about these things as techniques to try to understand why or how these genetic aberrations happen, how we can fix it, how we can isolate which of the genes that are implicated. So genetic engineering involves either adding or removing genes from a genome. We typically will do this in species other than humans, or modification of a gene. There's a number of different approaches. We can do selective breeding. So if we know, for example, there's lots of lines of rats or mice that have been selectively bred for a given trait. So we have something, for example, that's called the spontaneously hypertensive rat, that after a certain age, these animals suddenly start developing hypertension, which is high blood pressure. So if you want to study blood pressure, it makes sense that you want to try to selectively breed in this trait. So you make sure that you breed it with another spontaneously hypertensive rat and keep that gene in the gene pool. Of course, there's other problems because you're mating cousins with cousins, right? So a little bit problematic. Maintaining spontaneous mutations. That can be another way of selective breeding. So for example, that spontaneously hypertensive trait would have been a spontaneous mutation, so we can maintain that with selective breeding. You can directly manipulate genetic expression. So you can do some really funky techniques. And again, if you're interested in genetics and neuroscience, John Stead here in the Department of Neuroscience is our genetics expert, and he does things like this, where you can, and also Alfonso Abizade, who studies obesity. You can go in and, with some fancy equipment, you can change which genes are expressed, and as a result, see how that influences the behavior of the animal. Dolly the sheep here pictured is an example of an organism that was successfully cloned, i.e. the identical gene was used to produce yet another Dolly. And so you produce these offspring that are almost nearly genetically identical with another animal. You can imagine that this has some serious implications for the future of us as a species. You can use what are called chimeric animals, which are crazy things that essentially, like this is an example of a chimeric animal. an animal that has genes from 2 different species. Wacky, right? So this is a liger, is that what it is?
Yeah, it's like a lion and a tiger.
A lion and a tiger.
Apparently there's a tiger where it's like the difference between a male, it's like a male tiger and a female lion, and then a liger is like a male lion and a female tiger.
Right. So, yes. So you can get some crazy things happening. You can breed monkeys with chickens. No, you can't. I'm kidding. Transgenic animals are a gene that's added to the genome and passed along and expressed in subsequent generations. So if that particular organism doesn't normally have that gene, you can put it in to that animal. That's called transgenic, and have that animal express that gene and pass it on to future generations. The opposite to that is something called knockout, where you can take a specific animal, and this is the most common form of genetic engineering. If you want to see if that gene is important in feeding behavior, you can selectively knock out that gene and see how that influences feeding behavior or sexual behavior or motor function or whatever. Have an amazing weekend. Stay wet. I mean, dry. Stay dry. Don't stay wet. We'll see you Wednesday.